Good morning. It's a great pleasure for me to introduce you Professor Marcel Jaspers. Marcel is Professor of Organic Chemistry at University of Aberdeen since 2003. He was a lecturer and senior lecturer in Organic Chemistry at University of Aberdeen between 1995 and 2003. And he did his postdocs at University of California and in Texas. His research is mainly based on the discovery of new compounds with high biological activity from marine and extreme sources, and is interested in their structural elucidation and biosynthesis. In 2010, he founded the Interdisciplinary Marine Biodiscovery Center to focus on marine bioresources for novel pharmaceuticals. And from 2012 and 2017, Marcel led the EU FP7 funded project Pharmacy, interested in pharmaceuticals from marine microorganisms. And it was a project of 24 partners from 14 countries. And the output of this project, which ended a few months ago, were 5 compounds with the activity against epilepsy and potential activity for Alzheimer's disease. In fact, they worked also on animal models. But we will learn more during the talk. So thank you for accepting our invitation.
Thank you. So I'll, that needs to be turned off now. Thank you very much for the invitation to come and speak here. It's always nice to be back in Napoli. Wonderful food and people. So I'm glad it's sunny today after the rain yesterday. And also it was difficult for me to get here this time, so I got lost on the way and everything else. But first of all, today I'm going to introduce the research center that we have. And then I'm going to tell you a story that started in 1996. And it's a story of biological discovery, genetics, chemistry, and biotechnology. So it takes the whole story from the beginning to the end. So you have an idea of how such a long process can take place and what the benefits are of studying a single system for a long time. So first of all, where is Aberdeen? So it's in the north of Scotland. It's far away. It's cold, dark, damp. No, it's not so bad. So you see the picture at the bottom there is the King's College, which was founded in 1495 by Bishop Elphinstone, who felt that the people in North Scotland were ignorant and they needed to be educated in a proper way. So he founded King's College with the grant of King James at the time. And at the time, Scotland had 3 universities and England only had 2. So the Scottish are very proud of this historical tradition of education. For some time, Aberdeen had more universities than England put together. So it had 3 universities. Now it only has 1 big university. We have an ancient chair in medicine, and we also have the very first chair of chemistry as a separate subject from 1793. And again, only in the English-speaking world, because the Italians had it all first. The university is about 13,000 students and 3,000 staff. So it's not a big university, but it's reasonable size, and it has speciality in medicine and law. So our research center, its aim is, as Chiara said already, the discovery of drug candidates and research tools from extreme environments. And in order to do that, we need a lot of people. So what we have is, first of all, the biological facilities, biological organisms that we're interested in. Particularly, we started off with invertebrates, so I'll talk about invertebrates today, particularly these kind of organisms here, which are the sea squirts. So I'm talking about the invertebrate, and mainly it's symbionts. But then we started working predominantly on marine microorganisms. So that's the Pharmacy project came out of that idea of looking at deep sea and cold-adapted marine organisms, which is where the extremophile stuff came from. And working with good microbiologists also led me to work on desert bacteria. The people that we have, me as a chemist, Dr. Abel, Rainer Abel as a pharmacist. Again, many of you will know him already. Hai Deng also will be known to the audience here as a molecular biologist, a speciality in polyketides and non-ribosomal peptides. And Wael is my colleague who does a lot of the work that I will describe today. He is a senior research fellow. He runs a semi-independent research group in this field. And we have analysts, Jörg and Eva, and Laurent is a synthetic chemist. So less of that today. So ideally, we'd like to work on the reasons that these molecules are generated by the organism. And that's very difficult to work out. But so we talk about symbiosis, we talk about the relationship between an organism and its bacterial symbionts. We talk about biosynthesis, about why these organisms are making these molecules and how they're making them. But often, to pay the bill, we have to do applications. And most of our applications are in drug discovery. So particularly cancer, infection, inflammation, and parasites. And as Chiara mentioned now, epilepsy and Alzheimer's disease as well. The tools that we use are the tools of all natural product chemists, a lot of chromatography, a lot of spectroscopy, but now the tools of molecular biology to predict what we can find, some chemical synthesis to help us make substrates and test those theories, molecular modeling, and development of software for biotechnology applications. So we really, as this is just the profile of the lab, we're interested in deep sea, cold, and desert organisms. And again, the desert organisms came from this relationship with two very eminent microbiologists called Mike Goodfellow and Alan Bull, who were advisors on the European project Pharmacy. And they gave me bacteria from the desert. So I will be finishing a project on the Atacama strains in the near future. And we have some great scientists visiting at the moment from Chile. So we have two postdoc and a PhD working in our lab. And again, we're always interested in new biology. And the more interesting biology we get, the more interesting chemistry we get. The two things translate. So we're very interested in things that are new structural families. So you see these molecules here as a big picture of different types of molecules. And that will have associated biological activity. So the interesting part is that the molecules that are active against Alzheimer's and epilepsy are not new. They're old molecules with new activity. And even so, that's already very interesting for us. So the lab itself is a reasonable size. It's too small for everyone that we have. It's maybe 300 square meters. There's enough room for 10 people in there, in a chemistry lab, except we have 18. So we have to share very nicely. We have a molecular biology lab here, a microbiology lab. We have a spectroscopy lab containing a lot of big equipment. Then we have a separate lab for really high-end molecular biology. So rather than doing all of the work in here, in the top lab here, we have a separate suite of labs, which I don't have a photograph of. And they are, they have all the facilities for protein expression, cloning, bacterial culture, etc. So this is a nice place to work in. It's interdisciplinary relationship between the science, biology, chemistry, and analysis all at the same time. So the lab is very interdisciplinary, and a lot of people from different countries. So the reason I show this picture, I think this is our record audience of people, this a year ago. But because natural products is so international, we have scientists from all over the world visiting or working with us as PhD students. So again, we're very keen on what we call capacity building. So we have scientists who are now in Egypt, in Ghana, in Bangladesh, who have productive research groups working in natural products of their own biota. So I'm going to tell you a story that starts in 1996. And the reason I want to tell it is because it started off in my first year as an academic. My old professor, Phil Cruz, says, you know, you can come on one expedition with me whilst you're a young academic. You can pay for everything, but you can still join my research cruise in Manado in Indonesia. So we went here, and that year we went eastwards to Halmahera Island. So it's a beautiful place. It's one of some of the most beautiful research reefs I've ever seen. And this is one of the locations. These are from old slides, because of course there were no digital photographs, so it's not a great photograph. But you can get the impression that it's a very beautiful sort of tropical place. And Phil was my professor, and he's a mentor to me. He taught me how to do everything that I know. So he taught me how to run a research group, to write papers, to review papers, to write grants, review grants. He still helps me nowadays, and we wrote a textbook together. So he said, I've got some advice for you. He said, you've got a background in bioinorganic chemistry, or inorganic chemistry, so you should study metals in marine invertebrates. So I thought, this is a good idea. But nobody tied the two together, so we did. And it took me a further 11 years to get any publications on that topic, because it's really tricky to do. But it was a good idea. So this is him in Indonesia. But the second one was a chance discovery. So this is Miranda Sanders. I went to visit her in California recently. She was our taxonomist. A the time. She organized an expedition, and she said, you should collect this organism. It says it has some cyclic things in it. She could remember from her taxonomy and from her study of the chemistry of this organism that I should collect it because it contains cyclic peptides. And I was interested in molecules that could bind to metals. And I thought, maybe this is a good idea. So I did, and that's the first collection I made. So Lysocline patella is a sea squirt. It's a Indo-Pacific weed, essentially. It grows everywhere. It grows between July and December, and then it dies and disperses, and then it grows again July to December. So you have small bits in July. And it contains, it's a colonial ascidian, so it grows together. So it's a whole bunch of zooids growing together. And it contains blue-green, or alga, or a cyanobacterium called prochloron. So it's rather wonderful. You can get tens of milligrams of compound. So if you isolate this, this is very productive. And I learned from Michael Kuhl recently that the genes responsible for the production of the metabolites are the 4th most expressed genes in that organism. So they're really important for the organism. We don't know why, but we know they're important. Right? So they're important. The other thing that's interesting is that the symbiont makes a sunscreening pigment called microsporine-like amino acids for the host. So it exports it to the host. It's contained in the host. It screens out the harmful UV radiation for the symbiont. So is it selfish, but it exports it. It's really funny. So if you want to get a good source of microsporine-like amino acids, you would go to this organism and collect the host, and you extract it. Very high content. So there it is. There's the prochloron. But we knew, in my perspective, this is the cyclic thing that Miranda referred to. And those compounds are interesting at the very beginning because Bob Jacobs and Bill Fennikel showed that if you used it against multi-drug resistant cancer cells, you could reverse the drug resistance. So that was very interesting. So once you add a little bit of this material, so it goes from 90 nanomolar activity for resistant cells down to 1.5 nanomolar with the addition of a small amount of patelamide D, which is this compound here. So again, it does so by inhibiting the protein efflux pump. So normally does when you have a toxic compound going into a cell, the cell starts to express the export pump, and it will export the toxic compound into the environment rather than killing the cell. So this is one of the interesting parts. So we started working on this compound. At the beginning, so just for those who like phylogeny, originally there was a book by Ralph Lewin at Scripps Oceanography deciding that prochloron was a unique organism. It was all by itself. But then genetics came around, and genetics says actually it's just a cyanobacterium. Because nobody could grow it, and nobody still is able to grow this organism without the host. So it's, again, there it is, prochloron, and if you look a little bit more close up, it sits right in between other cyanobacteria, Trichoderma, Anabina, etc. So it's not so different. But initially it was thought to be very different and unique. It's just to show that it is sort of in between. And we'll see later on that knowing that it's part of the normal cyanobacteria enables you to find related compounds in other cyanobacteria. So the cyclic things, we published a lot of this new compounds in the beginning, so new chemistry, but we're also very interested in how these molecules were shaped in 3 dimensions. So that was our work in the early 2000s. So again, that technique took very powerful computers to do. It took several days to get the calculations to work. Nowadays, it's like that. Within minutes, you can do the calculations because, of course, computers are so much faster. That was interesting, but what I was really interested in, did these molecules bind to metals? And they did. So we did a lot of kinetics type work and a lot of binding studies. And what you can see is that the molecule is designed to bind to copper. You can do competition experiments to show that copper is the metal of biological relevance. And you can show that it only binds 2 equivalents and then stops. We didn't know why. We proposed many theories. And many years later, Peter Komba from Heidelberg in Germany phoned me up and said, I think we've proved your theory. So he came to visit us, and he has some beautiful studies to show that the reason that it binds the 2 equivalents of copper is because it acts as a catalase, not a catalase, carbonic anhydrase. So it acts to recycle carbon dioxide. So it may be involved in the host-symbiont interaction. So that's the next thing. So Michael Kuhl in Denmark and Peter Komba and myself are trying to think about how you can do experiments to prove that theory in situ. So it's a very nice possibility to study symbiosis. The other thing I was interested in, the molecules were interesting structurally. Just basically on structure, you could see how the molecules would fold in a funny way. And that in itself is very basic science. There's no application at all. And the result of that, I thought, I would look at these molecules, and you could get these children's toys that you use to show how the molecule folds. And just as a side shoot, I developed a teaching kit with an American toy maker to show people how to build proteins. So if you want to buy this, you can go to the web. So it's just a little plug. No, it was really to teach our first-year chemists how to understand protein structure. But it's a nice thing to have done, this side shoot. But let's go back. So then, after all this work on the binding, we decided we wanted more material. And I met a colleague from the University of London School of Pharmacy called Paul Long. And he said, shotgun cloning is your way. So if to get more material, you don't want to do synthesis, it's too difficult. But you want to be able to use the genetics to make more of this molecule. So we decided that we would do a joint sabbatical. So Paul spent 3 months in the summer of 2004. I spent from July 1st to December 22nd in Australia. My family came with me. And Ames is just here, on Cape Ferguson, just south of Townsville. And the Australian Institute of Marine Science is the premier institution for reef studies in Australia. It's on the Great Barrier Reef. And they have access to some of the reefs that are restricted, so only scientists can go to these research reefs. So the idea was very simple. What do you do? This is the design experiment. So Paul came to me. This is very early. So Joe Handelsman had shown you could do this shotgun cloning with genes from the soil, from metagenomes. So Paul said, what you do is you take your symbiont cell. We think it comes from the symbiont, so we're going to use that. But we can also use the host cell. We get the DNA. We put it into in this case, a back vector, bacterial artificial chromosome. We put it into a heterologous host, which is E. coli. Then we make a number of clones. We made, in our case, 1,500 clones, roughly, containing different DNA inserts from the host DNA, from the cyanobacterial DNA. And then you have to test which one is correct. So we said we'll do some kind of screening. We're not quite clear how we're going to do that. We're going to use DNA screening to start with. And then you scale up the productive clones and extract the compound, and you get the structure. So this was all wonderful. We thought this is going to take us years. Right? The Americans had tried it for years and years and years, and they had no results. They had tried a different methodology. This methodology worked in 10 weeks. We had all the results they would needed before the summer ended. It's crazy. This is totally unexpected that it would work so easily. And I'll explain why it worked in a minute. So there's lots of reasons why we chose a good model system. But first of all, so we could always find a compound. It's always there. And now that we know from Michael Kuhl that you can get, it's the 4th most active gene expressed in the organism, it's going to be there. You're going to find it. Okay? We could get to the reef. So they had done, before we arrived in Australia, they had already been to the reef for an expedition. They had collected the organism, they probed, and the compound was there. So already they did some preliminary work, so it was really helpful. We predicted that the compound was produced by the symbiont and exported to the host because of the way it should be formed. So the biosynthesis was, at the time, thought to be a particular type, a non-ribosomal peptide. It turned out not to be that, but at least it gave us a target. So then it was easy to extract DNA. We knew that already. Somebody had done it. Oh, so yes, so fine. So we went to the reef. So this is the Lady Boston, the Ames research vessel at the time. They have a much nicer one right now. But it can sleep 7 scientists and 5 crews, so it's very small. It's a very shallow draft, so it can go out to the reef. It doesn't hit the reef. And it would locate itself in the reef. So we went to Davies Reef, which is, again, classical reef structure. The prevailing wave action is from this side. The ship has a permanent mooring. The vessel has permanent mooring here. And there's a weather station here that can report whether the weather's high. Of course, this time it's July 2014. And we do some collections here, and you find most of the organisms are growing on the back of the reef where there's high current action, lots of water, lots of food in the water for the sea square to live off. And it took us 2 days to find the organism because we didn't look properly. But then we found lots of it. Even if it's July, when it's very small, you can find lots of organism. So collection is always nice. Okay, I don't do much diving anymore, but that was water collection is always wonderful. So the organism itself is very easy to handle. So this is my colleague Walt Dunlap is holding the organism. And Walt was the guy who dedicated his ship time to us. And you see the organism there? It's because it's an ascidian, it contracts when it is collected. You can squeeze it to get the prochloron out. It's so simple. It's just in the cloacal cavity, so it's in the layer between the organism and its waste stream. It sits there. You squeeze it, and you get a tube. So this is the body of Paul Long, my colleague. Okay, you'll see him in real life. But that enabled us to get very clean prochloron very quickly. So you just centrifuge and sterile seawater, resuspend, and centrifuge again, and you get 99.9% prochloron cell. So it's very simple to do. I did the chemistry, so we had an HPLC on the vessel. So as soon as the vessel is moored, you can construct everything. And we had standards, so all we had to do was to prove that the compounds were in the organism. So we did. So it was very simple to show that the whole organism and both the prochloron contained those compounds. So it's easy to do. So you just do standards. You can do quantitation quite easily. And it's very clear that the compound is there in large quantities. It's very, very good to know that when you're on the ship, you didn't have to go back to shore, do the experiment. You can do it all in situ, which is the really positive part of this experiment. So then Paul is extracting the DNA. Once we know the chemistry is there, we said this is a good sample. He's doing a DNA extraction. Again, the DNA is within 2 hours of collection. So it's very fresh. Okay, at this point, it takes 30 minutes to do the HPLC, extraction HPLC, so it's quick. At this point, Paul is asking, where's the hot water bath? He needs to do an incubation at 37 degrees. And Walt, the expedition leader, says, oh, there's no hot water bath. He didn't ask for one. And we're 6 hours from shore, so we have to make do. So what we do is we stick it to the engine, and the engineer turned the engine up to the right level. So that's about 37 degrees, and it's agitating. So again, the scientific paper says 37 degrees with agitation. It's pretty good. So then Paul is showing a shocking disregard for safety. He's doing the phenylchloroform extraction, so old-fashioned DNA extraction. Okay, it worked really well, but he should be wearing safety glasses, and he knows that. And I show this picture every time and say, bad safety protocol. But we got really good quality DNA. And it was long sections of DNA. So we were able to show, we didn't have pulsed field gel electrophoresis, but we were able to show us that by comparison to lambda DNA, I should say not LDNA, we could show that it was indeed very big DNA sections that we had. So we could say that the predicted pathway was 30 kilobases, and these were more than 50. So we could say that if we did the generation of the bacterial artificial chromosome, we would have the pathway captured somewhere as a whole. So that the average chance is that we would find that. So we could show, this is our vector, which came from Italian company, Donadio. I can't remember what the company's called. Stefano Donadio gave it to us anyway. So it's a very famous vector. But it has a number of features that make it very, very successful. It has kanamycin resistance, again, so you want to grow it. You grow it with kanamycin, and then you know that the cells that survive have the vector. It has insertional activation of the sucrase gene. So if your DNA goes in there, it disrupts the sucrase gene, so you can feed it with sucrose and it doesn't kill itself. So it's again very handy. And we know that it was prochloron DNA that was in there, and we know that it was most of the constructs had it in. So we had 1,533 clones with different bits of DNA. So the question then is, how do you screen for it? So 1,433. So the problem was that when we looked for the most likely way it was produced, which is non-ribosomal peptide synthase, everything failed. And the Americans had found the same thing. So then we thought, well, let's go back to a simpler method, and the simpler method is chemistry. So again, this is 2004. We didn't know about the ribosomal pathway, but what came out of it, that we were able to do a very simple deconvolution strategy. So first of all, we used the analytical methods to show that we could get the level of detection that we needed. So we grew the different clones in a 1.5 milliliter culture each. In all of these wells are different clone. But then, rather than testing each one, we combine everything. So it seems like a bad experiment, right? You combine everything. The reason is that if you have a rare property like this, maybe 1 in 1,500, then you can do this. If everyone is expected to produce it, then you wouldn't be so useful. So we did that. We combined. We found a positive plate, one plate. Then for the positive plate, we grow each row again, right? We combine each row, so now we have 8 combinations. And then finally, in each column, we grow each well, again, in large quantity, so 50 milliliters. And now we found a single positive well. Okay? 3,4G, I still remember the name. The plate 3, column 4, well G. It's really important. So to show that it worked, again, in comparison with the standards, so the standards are here and here, 2 compounds. And you can see that the retention time and the mass spec and the fragmentation is identical to the standard. So we had produced the molecule. The question is, why is it producing 2 molecules rather than 1? So again, that already indicated it was unusual. And we knew that at this time it was probably ribosomal, because there was another pathway that people are thinking about. People weren't aware of this. So the quantities produced are tiny. So 100 micrograms per liter, roughly. So you can just about see it, but it's not producing a lot. That was the exciting part, that we had done it. And this is the result after 10 weeks. So then we had to go further and do some more proof. But what happened at the same time is an American group had the group that hadn't been able to do anything. Good news of our results from a poster in California. And they had been working on the same thing, but they had some genetic information. They had the first draft genome of the organism. And they said, once it's ribosomal, we know that the starting sequences must be one of these cyclic permutations, right? Because it's a cyclic compound, you know it can start at any of these positions. First of all, you check whether that's a rare sequence, and it is rare. So therefore, when they found it, they were able to sequence up and downstream, right, and find it. So they were actually cloning a very large pathway. 11 kilobases at the time was difficult to do, and clone it, and then express. Now, they knew what we were doing. We didn't know what they were doing. They didn't contact us and say we would like to publish with you, which is a shame. They could have done it. But our work was the opposite way around. And the problem is that sequencing was expensive, and the one chance we had to sequence the genome failed. So we wasted a lot of money on sequencing that was pointless. So it was very, very sad. So we got published in one journal, they got published in another journal about the same time. So here it is at the bottom. So they got PNAS, we got Chem Biochem. But it's difficult to say. Anyway, the point is that once you look at the gene cluster, it's a very interesting one. It's very simple. It's a ribosomal one. And what you can show is that this one is the precursor peptide. It's the one that gets processed by the enzymes. And the enzymes that process it are part A, part D, part G. Part F is inactive, and part A and B, the genes are not necessary. So you can knock those out, and nothing happens. There's no change. You still get production of the compound. We don't know why, but we don't know what the other genes are doing. So anyway, what happens is this leader peptide is recognized by this enzyme here. So this is the heterocyclase, and the heterocyclase makes these little heterocycles. It makes oxazolines or thiazoline. Okay? It's a very complex process to do very simply. So a chemist would take 3 or 5 steps to make this same molecule. The biology does it really efficiently in less than 2 hours. It's incredible. And then what happens next is this side gets cleaved off. The molecule gets cleaved off here, and it gets made the round. The macrocycle is made, and then the oxidation takes place. We don't know. Sometimes we don't know which steps take place when, but we're pretty sure it's roughly that order. Yeah, so it's still difficult to prove. So those are great molecules to work on, and basically they have a number of features. So if you look at all of them, you get the feature of the heterocycle, you get the feature of the oxidized heterocycle, you get the macrocycle, you get the inversion of the stereocenter here, and you get the possibility of a prenyl group over here. So all of these enzymes are encoded in a single